Perfect. I'm Mariana Inés, and I'm the co-leader of BINA's Diversity and Inclusion initiative. This is my first time introducing this presentation in Spanish, so we're celebrating Hispanic Heritage Month for the first time, which is a whole month dedicated to celebrating the contributions of our Hispanic. So this is our first time working with the presentation of scientists, the contribution of our scientists to the community. And so they are obviously valuable members of our community. They have a lot to tell us about career development. So for the wonderful participants who are valuable, gracias. We will be listening to the career, we will be hearing about the career and trajectory of Pablo Ariel from the University of North Carolina at Chapel Hill. He did his master's degree in biology at the University of Buenos Aires. He also focused over here on synapses. Sorry, folks, the interpreter's a little distracted. And also later made it to become staff scientist at the research center at Rockefeller. And now he is the associate professor and director of microscopy services at UNC. He's also the author of a very useful, very detailed guide for the use of the vision. And most importantly, he's very well recognized by the scientific community as the director of microscopy. He helps users in several different fields. So as director, I am also very happy, very pleased to introduce Pablo, and I'm excited for this talk. I believe it will be very inspiring, and I'll hand it over to Pablo.
So thank you so much, Mariana, for the introduction. Yeah, I'm delighted to be here, and I'll be happy to take questions at the end in Spanish or in English, whatever you prefer. So with that, let me get started. Let me see if, there we go. Okay, so my job is to help other people with their research. So I don't have my own research lab, I'm a helper. I help other people with their research needs. And in this talk, I'm going to talk about three things. First, how I got my current job. Second, I'm going to spend most of my time discussing microscopy myths that I encounter every day in my job. And finally, I'll have some thoughts on certain aspects of my job that I think will be relevant to people who are curious about what the job entails. So how did I get my job? Mariana sort of went over this a little bit, but I'm from Buenos Aires, Argentina. I got my degree in biology called a licenciatura there. And then I came to the US where I was a graduate fellow at Rockefeller University in New York. Then I was a postdoc at Columbia, also in New York. And then I took a swerve in my career and I became a research support specialist back at Rockefeller University in the imaging core facility there. And I really fell in love with that kind of work and was very lucky. I got recruited to my current position as a core director, and I'm an associate professor at the University of North Carolina at Chapel Hill. So if any of you are curious about any of the steps in this progression, it wasn't as simple as this line suggests. I'm happy to talk about that at the end. One other thing I want to share with you that I've found very useful as I navigate my own career is I use a sort of Venn diagram that I'll put up as a guide. And so this Venn diagram has a few things. So there's this circle, which is what I need to succeed in a job. And there's a bunch of stuff in there. There's a bunch of stuff that I'm good at. And then there's a bunch of stuff that I like. And there are intersections and non-intersections of those three categories. And I always try to the degree possible to get jobs with as much stuff in the intersection as possible. Because when I'm doing stuff that intersects those three things, I'm happy. And if I drift away from that, I'm not. And so I'm trying to maximize my happiness in my work life by trying to get jobs where mostly I stay in the intersection of those three categories. I'd also like to just briefly mention that really luck was a huge factor in my own career. I happened to be in the right place at the right time so many times that it's ridiculous. So I was at great institutions with great colleagues and great mentors, and a lot of it was luck. And so, as advice, be lucky is not good advice, but I would say that, be in a position where you're prepared to take advantage of good luck if you get some lucky breaks that go your way. I think that can be useful. And also happy to discuss what that might look like more specifically with people at the end. So let's go to the sort of meat of my talk, which is to discuss microscopy myths. And so, a reasonable question that you might want to ask is, why should you listen to me? Like, well, so I'm coming from a place of experience. I don't know how this happened. I don't feel like I'm old enough to have this much experience, but somehow that did happen. And so I have almost 20 years of experience in microscopy, more than a decade in imaging core facilities, and I've personally conducted more than 1,000 trainings. So during the course of that experience, I've seen a lot. I've seen good, I've seen bad, and I've seen, quite frankly, ugly. And so there are these myths that seem pervasive in as I do my job. And so I want to share them with you and hopefully debunk them. And so the first myth, almost the foundational myth that I find, is that microscopy is not quantitative. So this comes up all the time. And I would say that I very strongly disagree with that. In my opinion, the best microscopy is quantitative. And I'm going to give you an example from my colleague Vidya Ragnaraju, who I was in Tim Ryan's lab with as a graduate student. She's now a group leader at Max Planck. And so Vidya was interested in measuring ATP concentration in live synapses using just images. So synapses are connections between neurons. And the way she did this is very clever. She attached a special reporter molecule to synaptic vesicles. Synaptic vesicles are subcellular organelles that localize to synapses. And the reporter molecule had two components. First, it had an M-cherry, that's a red fluorescent protein. And second, it had a protein called luciferase. So luciferase is a thing that takes luciferin, ATP, and oxygen, and in a reaction that turns luciferin into oxyluciferin, it releases photons. So this is the thing that's in fireflies and makes them glow. And it's been used by scientists all over the world. And so she took these two things. So she delivered both luciferase and a red fluorescent protein to the synapses. And then she took images in the fluorescence channel, which allowed her to look at M-cherry, and in the luminescence channel, which allowed her to see the photons that were being released by luciferase. And then she did something very clever, which is she took the neurons and she permeabilized them. So she made holes in the membrane and applied different concentrations of ATP to the media. With that, she was able to construct a curve of the luminescence by fluorescence ratio as a function of ATP concentration. Now, the reason she divided the luminescence by the fluorescence is to be able to calibrate for the amount of reporter molecule that was present in her preparation. And with this curve, what you have is something that allows you to predict for any ATP concentration what the L by F ratio will be. But more importantly, you can go in the other direction. You can make a measurement on a live neuron of the L by F ratio and determine what the ATP concentration is in that live neuron. And she went on to do this under multiple conditions and was able to discern what happens when neurons are active, what the contribution is of different metabolic pathways, et cetera. And I love this paper, even though, it's out of date now, there are better ways of doing this. But I love this paper because she measured an ATP concentration and all she did was take images and be super clever. So the best microscopy, in my opinion, is quantitative. And this is, I think, a wonderful example of that. So a second myth that I've run into over and over is that controls are optional. And you may think that I'm being exaggerating, but I can assure you I am not. I frequently get questions of this kind. Someone will come and tell me that they have done immunostaining for protein X. They show me an image and they ask me, is this green signal protein X? Is this real? And so I immediately respond by asking about controls, because simple controls can be very informative. And so if you've taken the image that I just showed you in a condition where you had a primary antibody against the protein of interest, a secondary antibody conjugated with a fluorophore that will amplify the signal of that primary antibody, in a preparation where you expect protein X to be present, and you get this image by itself, image doesn't tell you much. Because if you prepare another condition where you don't have the primary or the secondary antibodies, but you do have protein X, and you see something like this, what this is telling you is that the signal is actually autofluorescence. The signal that you see is not protein X. It is present there because the cells have things inside of them that fluoresce without you having put them there. So you need other controls to be able to determine whether that is the signal is coming from where you think it is. So for example, if you run that first control and you don't see anything, you can rule out that the signal is due to autofluorescence. If you do another control where you just add the fluorophore conjugated secondary antibody and you see signal again, what this suggests is that what you are seeing is because of non-specific binding of the secondary antibody to your sample. If you take this one step further, you do these controls that I've mentioned and you don't see anything, and then you do a final control where you put on the primary antibody, you put on the secondary antibody, and you don't put in protein X or you remove it somehow, and you see a signal, then that means that everything you saw was due to non-specific binding of a primary antibody. So this is just illustrating that simple controls can be very informative, and yet for reasons I don't really understand, they're often ignored. So this is a myth that these controls are optional, that I run into quite frequently. So if instead, you do all your controls and you don't see anything in the places where the control should give you negative results, then you can be fairly confident that you are looking at protein X. So let me jump to another myth that's related in some sense to the previous two, and that myth is that statistics are optional. So what do I mean by this? Why am I talking about statistics and I talk about microscopy? So this comes up in two ways. The first way is people frequently ask me or I ask them, where in a sample will you take images? And then I ask them, how many images will you take? Or sometimes they ask me these questions. And so these questions have to do with statistics. So if we look first about where in a sample will you take images, when I ask folks these questions, this is the kind of answer I get. Well, I'll look around for representative cells. And so my question is, how will you look around? And how will you know that the cells are representative? And so there's a better answer to how to do this, which is random sampling. So you just set places at random to look, and you can do this with most modern microscopes. And so when I propose this as a solution, people give me a very reasonable response, which is to say, well, I don't want to waste time taking images of dead cells, of dying cells, of rounded up cells, of cells that are too big, of cells that are too small, of cells that are overexpressing the protein that I'm interested in, or that they don't express it enough. And so my answer to that is like, that's fine, that's reasonable, your time is valuable. You can do random sampling and then just add clear and disclosed exclusion criteria. So what do I mean by this? And sort of how do we illustrate this? So if you imagine that you have a population from which you're taking a sample, and then from that sample, you are measuring something and making inferences about what's happening in the total population from whatever you measured in the sample. If you have exclusion criteria, what that means is that your conclusions apply to a smaller population because you've excluded from the population that you're sampling from cells that are too big, overexpressing, dead, too small, or whatever it might be. And so as long as you're clear that this is what you're doing and the population that you're making inferences about is a population of interest, then this is fine. And it's much better than just looking around where you could have biases that you're not even aware of. So there are some exceptions that actually prove this rule, which is if you need to know whether something can happen and not how often it happens. If you want to show that something can happen, then you just need to show it once, because that is by definition an example of it happening. And so an example of this that I was involved in was uh, pathologists in my department were trying to diagnose a sample with Chagas disease, and they were looking for specific structures called Trypanosoma cruzi, that's the organism that causes disease, mastigotes, in a human heart biopsy. And so if they found those things, then they knew that that person had this disease. And so they were just looking for those things. They weren't looking randomly. They were just looking all over. And once they found it, they were done. So this is a case where, you know, you don't really need random sampling. You just look for the thing. And if you find it, then you know that they have the disease. The other kind of question that comes up that's related to statistics is how many images you will take. And so when I ask folks these questions, I often get an answer that looks like this: 5. And so I asked, okay, why 5? And the answer is like, well, you know, a postdoc in my lab told me to do that, or someone in my lab told me to do that. And that's really a terrible answer. And luckily, there's a better answer, which is to use something called power analysis. So the idea here is you have a population that you're interested in, let's say all HeLa cells that could exist in the universe, and you take a small pilot sample. So you take some images of a small number of those, and then you measure something. So let's say you're interested in cell size, okay, like the area that they occupy in two-dimensional space. So you measure that thing and you estimate the variance in that measurement. Then you decide how big of a difference in that thing that you're measuring you're interested in, and how big of how big of a probability of making two types of errors you're willing to accept. One, finding that there is a difference when there actually isn't one there. That's a type 1 error. Second, not finding a difference when it is there, which is the type 2 error symbolized by this beta parameter. So if you have all this information, you can put it together on these very simple power analysis calculators to get an estimate of what the size of your sample needs to be, so which is here symbolized by n. And this will help you avoid wasting time and money. If you take samples with too many, do you take too many samples, excuse me? This will cost you a lot of time and money. If you take too few, you won't be able to conclude anything with any kind of rigor. So that is sort of myth number 3, statistics. So now I want to go into a myth that I've run into quite a bit as well, and that is very much in the weeds of microscopy, which is that colocalization analysis is easy. And so I've run into this in almost comical ways sometimes when I have a discussion with someone about their experiments, and in the last 5 minutes, they say, oh, and can you just show me how to do colocalization analysis? And I can't do anything but laugh, because the reality of colocalization analysis is more like this. So when you're doing colocalization analysis, you're facing this labyrinth of decisions in a murky environment, and there's no clear guidance in which direction to go to. So I want to dig in a little bit more to illustrate the many decisions you need to make, and how at each step you're adding uncertainty. So there's a foundational issue, which is, you know, what are we even talking about? Colocalization as a word is very much a loaded term. There are many different things that people are referring to when they use this umbrella term of colocalization. And in the literature, there's been this, I think, reasonable classification of the things that people are talking about into 4 categories. The first is co-expression, which means 2 markers are present can be present, excuse me, in cells, but not necessarily in the some cellular compartment. Co-occurrence, where you're interested in whether 2 things can be in the same subcellular compartment. Correlation, where you're interested in whether the concentration of one of the components is predictive of the concentration of another. So you can imagine, if there's more of one thing, does that mean that there's more of the other? Maybe because they're interacting. And co-distribution, which means for techniques where you can determine with high precision where individual molecules are, can you look at the pattern of distribution of those molecules and say something about whether those patterns are more associated with each other than what you would expect by chance? So in the rest of today's talk, I'm basically going to focus on co-occurrence and correlation, not because, the others aren't interesting, just because this is what most people refer to when they're talking about colocalization. And so there's a key thing as well, which is to interpret this colocalization concept, you need to have an idea of resolution. So the level of fuzziness in an image, the level of discernment that you have. Because if you imagine in one extreme, if you imagine the entire universe were in a single voxel, so a single three-dimensional pixel, then everything will be colocalized inside the universe. If you imagine the other extreme, the atomic level, so this is an image of the spike protein of COVID binding to fragments of antibodies. So those things are interacting. We know that they're interacting, but that doesn't mean their atoms are overlaid with each other, because that's not possible because of something called the Pauli exclusion principle. Two things cannot be in the same place at the same time. So really, at the atomic level, nothing is colocalized. And light microscopy is somewhere in between. And different forms of light microscopy are on different parts of the spectrum. So if you look at the far left side of this image, we have an image of the nuclear pore complex, lamin, and DAPI. And you can see that the nuclear pore complex, which is in red, is overlapping with lamin, which is in green. So that's with confocal laser scanning microscopy. If you look at this same sample with another technique called 3D structured illumination microscopy, which has higher resolution, albeit not atomic resolution, you can see that the nuclear pore complexes are actually not in the same place as lamin. They're slightly offset from them. So, as you look with different techniques, you can see different degrees of things overlapping or not, depending on the detail with which you look at them. And so you can imagine standard microscopy giving you this sort of blurred view of things. And as you increase the resolution through the use of super resolution microscopy techniques, a lot of things that seem to overlap and that are shown here in this sort of yellow are actually just near each other, but not actually overlapping. And the key point is that colocalization doesn't not imply interaction. At the molecular level, there is no such thing as colocalization. And at best, what you can see is whether two molecules are within a certain distance of each other. So the analogy I always propose here is we can determine if two people, for example, are in the same room, but not whether they're shaking hands. Now, if you're interested in whether they're shaking hands, it is a necessary condition for them to be at least in the same room, but it is not a sufficient condition. So colocalization does not imply interaction. It just implies things are within a certain distance of each other, given by the resolution limit of the optical system being used to measure those things. So let's assume that you know what you mean by colocalization. You don't have any confusion about what you're talking about, and that you understand the effect of resolution, and that you're going to acquire good data. What I mean by good data is data following a bunch of practices that I don't have time to go into today, but they're all over. I think maybe the interpreter leaked to the other channel. Hopefully that's been corrected. So are we good to continue? Okay, thanks. So let's assume, again, you know what you mean by colocalization, you understand the effect of resolution, and you acquire good data. Then what? Now what do you do? So a typical thing that people do is use visual approaches that unfortunately are mostly useless. The typical way those show up are in plots in images where you see one channel in red and the other in green. A more colorblind friendly version of this is to look at one channel in magenta and the other in green, where the overlap shows up in white. And so you see things that look something like this. This is an example with two proteins, one called STING, the other one called NLRC3. And you can see different degrees of overlap of the proteins in white. But you might notice this is actually the same image that I've just tweaked in different ways. And so this highlights the problem with this visual approach, which is it's very easy to manipulate by adjusting the contrast of the different channels differently. And it's really hard to interpret anything you see outside of very obvious cases where, you don't really need to measure anything because it's so obvious. But for most things, this approach does not cut it. You actually need to measure something. And then the question becomes, what do you measure? Well, what do we measure? Because there are many different metrics that you could use. Now, there's a category of thing that you can measure, which are called pixel-based, because they're based on the actual pixels of the image. And there's sort of three categories within that subfamily, which is you can look at correlations between the intensities of the pixels. You can look at the fraction of intensity that overlaps, or just the overlapping amount of the pixels that you consider to have signal. Or you can look at intensity cross correlations by shifting one image from the other and seeing how correlations break apart. So that's a sort of a certain category of metric that you can use to estimate colocalization. There's another category, which is you take these raw images and you do something called object detection. So you do an abstraction of the raw data and replace it with objects, for example, dots or spheres. And then you make measurements based on those objects. So that's why they're called object-based metrics. And you can, for example, say that you're interested in the distance between these. And if the distances are closer than a certain amount, you may think that things are colocalized, and the same with sort of volume or area overlap. So you will need to make a decision about this. And it's not always clear what the best option is. Once you've decided what to measure, there's a key step, which is you need to separate the pixels that have actual signal from the ones that you consider are in the background. And so this is deceptively complicated. The idea is you typically set some sort of intensity threshold for pixels. So you will only include pixels in your analysis with a certain intensity. But how do you find the right threshold? So how do you find that intensity above which you include pixels? And so one option is to do this manually. So if you do this manually, the approach is you're going to try different threshold values. And for each threshold value, you're going to see which pixels are included or excluded. And the problem with this is there's no way to know which threshold is appropriate. It's completely subjective, and scaling this to many images is actually quite hard. So what is another approach that we can take? So another approach is to do this automatically. So what does that mean? So what that means is you have to pick an algorithm to do this for you. But there are many different algorithms that you can select, and this just shows the results of applying those algorithms to the previous image. These are just the 16 basic options in Fiji, which is a very kind of well-known image analysis software that's open source and free. And so you have many, many different options. And how do you decide which one of these you want to use? Again, there's no easy way of doing that. Another issue with this colocalization analysis is how do you keep this analysis consistent across many images? So there's a couple of ways that you could do this. One is to use the same automatic method across many images. The other, if you're using manual thresholding, is to use the same manual threshold across many images. If you can't use the same threshold on all the images, then you're left in a kind of complicated scenario where maybe the solution is to have the same person doing manual thresholding across all the images. And each of these options, has pros and cons. And a priori, like, you will have to make a decision as to what to do. There's no right or wrong way to do it. It depends on what your data is like. Another issue is, let's say you make these measurements and you get some numbers. What do you compare it to? So the best option, in my opinion, for what you compare these numbers to are different experimental conditions. So examples of this can be biological, positive, and negative controls, proteins that we know do or do not overlap in the cell, drug treatments, or the passage of time. So all of these are things in the experiment that you can do where you have different expectations for the degree of colocalization that you're going to have. A much worse option is to compare your results to some sort of randomized image. And there are different ways of randomizing images. One is to rotate the image of one of the channels. Another is to randomize all the pixels. Another is to randomize the pixels, but in groups that are the size of a point spread function. Therefore, they're groups of correlated pixels. And finally, you can do that, but within certain regions of interest in an image that typically correspond to a cell. So let me show you one example, which will illustrate why these are a little bit difficult to really be sure that you're doing the right thing. So this is the example of randomizing by rotating. So this is the original image in the two channels, and the third image, you can see the overlap in white. So the idea is you measure some colocalization parameter in that data set, and then you take one channel and you rotate it. And then you measure the colocalization parameter with one of the channels rotated. And the idea is that if the colocalization is real, you will expect a lower colocalization measurement in the rotated condition. So that's the idea. So it's like, oh, if the number's lower, then that means the colocalization was real. The problem is that when you do these kinds of rotations, you end up placing signal from one channel in places where it makes absolutely no sense for it to be here. So placing the signal of the magenta channel in that region that I've highlighted makes absolutely no sense. Even if that protein were distributed randomly, it would never be outside the cell. And this is an exaggeration, obviously, but actually most randomization schemes have some version of this problem because you can never be sure that they don't underestimate the overlap in this randomized condition. So I really don't like comparing to a randomized image. I always prefer comparing to some sort of biological control. Now, once we make all these decisions, and there are more that I haven't gone into, but once you make all these decisions and analyze your data, there's a key question, just like you made a lot of decisions. How robust are your conclusions to slightly different decisions? So how do you know that if you hadn't tweaked something a little bit differently, your whole result would not fall apart to pieces? So I'm going to give you an example from a collaboration with my colleague here at UNC, Jenny Ting. And so, Jenny's lab came to me and they had the following sort of simplified results suggested from biochemistry. They had two proteins, one which I show in red and another in green. And in the presence of another thing, this was a piece of DNA, those two proteins, it seemed like their interaction probability went down. And so we did an imaging experiment looking specifically at colocalization, and we made many decisions for how to do this experiment, and we ended up measuring, so that red one is the STING protein, the green one is NLRC3. We measured the percent of NLRC3 signal that was colocalized with STING, and we found that in the absence of that thing that we thought would make them not interact anymore, we got on average colocalization values that were higher than when we put in the thing that supposedly kept them from interacting. And so this was good evidence that the biochemistry was onto something real. We went further, not in the paper, but in the paper we did a sort of crude version of this that we didn't even include in the paper. But I later went in and did a deep dive into the data where I said, okay, like the way we in particular did this is we established thresholds for pixels to include from channels 1 and 2, which I think they were called actually 3 and 4, or 2 and 3. And so what I did is, if you look at this graph is a graph of the threshold in channel 3 versus the threshold in channel 2. And then it's a surface graph where the intensity of the color gives you a sense of how different the two groups are, with and without the thing that causes these proteins to cease interaction. And you can see that let me see if I have like a pointer or something here. Yeah, so you can see here that this this little crosshair that reflects the actual image analysis thresholds that we used. But even if we had changed those by up to 25%, we would still be in a region where this condition would be higher than this condition. Now, if you look at the p-values of the comparisons, that's also true. We could have changed the threshold by up to 25%, and the p-values would not have really risen beyond this arbitrary 0.05 threshold of statistical significance. So all of this is to say that the results were very robust to changes in the thresholds. And in fact, they're robust to all sorts of other changes of how you analyze them, which I've done later. So if this is of interest to you, I've created a comprehensive workshop with videos, instructions, data from this paper, not the full data sets in 3D because it's too much data, but a simplified version of the data, macros and results that you can, it'll take you a few hours if you go through this. It goes into great depth onto how to make decisions as best as you can when you're trying to do colocalization analysis. So check that out if you're interested. You can get to it through my core facilities website resource page, or you can Google us on YouTube. I'm sure you'll find it. So another myth, this is, I think, the final one that I have to discuss today, is that less is more when reporting methods. So I run into this all the time. And so here's an example. This is a real method section from a real paper that was published in Nature this year. And so this is the extent of description of the imaging methods. That's what I've highlighted in blue, and of the image analysis method, which I've highlighted in orange. And they used 3 different microscopes, one of which was actually not in my core, even though it says it was in my core. And they actually performed colocalization analysis. And this is the extent of detail that was there. So my point is not that that the results are garbage. My point is that I have no way of knowing based on this description. This phenomenon, I'm not just picking on that one paper, this is a widespread phenomenon. So folks looked at this systematically in this publication. They surveyed 240 papers in high-profile journals, and less than a fifth passed a minimal reporting quality threshold. This wasn't even, explain everything in detail. This was, I think their threshold was like, say these 3 things, less than a fifth of the papers passed it. So to state the obvious, this is very bad because without details of the imaging and the analysis, we can't evaluate results critically and we can't replicate what anybody did. And it's very easy to do better than this. So, my website has explanations for what to report in materials and methods for the users of our core. But, my colleagues have published, papers on this in high profile places that give you a guide to what to report when you're trying to explain what you did in a microscopy experiment or in an image analysis experiment. So it's really very easy to do better. And I think as a community, we should. So that's the end of the myths section. A few final thoughts about my job. So the first is, this is a service job. So if you have my job, you are a helper, and it's like you're in a store where you have customers that come and your job is to help them. Now, unlike in a retail scenario, the customers are actually not always right. And it's actually your job to explain if they're not right, because you want to avoid them wasting time and money. So it's really important that you don't think that just because they're customers, they're always right. They're not, and it's your job to help them understand when they're not so they can change what they're doing and not waste their time and their boss's money. However, customers are always right, in my opinion, about how they feel. So if they have a bad experience in your core, that is a problem that you need to address. If the bad experience was because of anything that's under your control. And over the years, what I've discovered is, this is maybe a trivial statement, but like a key skill is listening. People detest being treated like robots or interacting with robots. If you've ever called an airline or a cell phone company, you'll understand what I mean. If you get the computer, it's really annoying. If you get a person that treats you like they or you are a computer, it is also really annoying. So it is very important to listen to people. Just listening, even if you don't have a solution, will make people feel better and will allow you to walk towards where the solution might be. If people feel that you are disrespecting them by not listening, things will not go well, and it is very hard to pull a relationship out of the garbage if it fell in there because you are not paying attention to what they're saying and instead are, pontificating about all the wonderful things you know. So listening is really a key skill in this line of work. So if you don't like listening, if you don't like helping people out, then this is definitely not the job for you. Um, the other thing I'll say is that the impact in positions like mine really goes beyond papers. And the things that you do that maybe have the most impact are typically not papers, but they can be things like workshops, like the one about colocalization that I mentioned, videos, video tutorials, stuff you put on your website, or non-traditional publications. So I think, for example, Mariana mentioned this. So several years ago, I wrote a guide to an instrument that we have, which is a light sheet. I haven't talked about that in this talk. I still get emails from people saying, "Oh, thanks, I found that and it's really useful." So that clearly had an impact, but that's not a traditional publication because there was no way. That's not a paper. That's at most a chapter for a book that nobody asked me to write. But I just put it out there, and it's had an impact. And I don't know that it's had an amazing impact on my career in terms of bean counting of publications, but it certainly had a huge impact. So the impact in things like in these core facility positions really does go way beyond traditional uh, papers. Um, so I'll end by saying that I think cores like mine, so core facilities, they can teach critical and quantitative thinking that leads to better microscopy. And this better microscopy inevitably will lead to better science. So this is sort of why I do my job, and I'm happy. I feel like I am making a contribution in this sort of helper role by helping lift sort of the baseline level of competence of everyone when they're using a microscope or they're analyzing an image, and that's having an impact on the science that everyone is doing. So with that, I'm happy to take questions in English or in Spanish, and I'll stop sharing my screen.
Thank you so much, Pablo, for that fantastic talk. I think it's never too few times that one sees how the challenges and what is like the control issue that you discussed and all these myths. I think many of us have faced them. Hopefully not, many of us understand it better to not do it anymore. I don't know if there are questions in the audience. I didn't see any written so far, but I have a few for you. So one would be well, everybody gets warmed up. How was it for you when you chose to, like, did you ever wonder what do I want to pursue? Was it a natural decision? Were you always like, I want to work at a core and be a core director? Was that something always there?
No. So
Mhm.
the jump from postdoc to a position in a core uh, was not something that I had planned all my life. I was sort of uh, on this track that I thought would end up with me being a principal investigator in with a classical lab. But so if you think about the Venn diagram that I showed in graduate school, I was very much in the center, sort of in the intersection of all the things, and I was having a great time. When I transitioned to a postdoc, that completely broke down, and I realized that the things I needed to succeed were not the things that were making me happy. Uh, at all, or the things that I was good at. And so I very quickly was miserable and sort of looking for other other career opportunities. And in terms of being lucky, so that was definitely a place where I was very lucky. But I was sort of the thing I could say, like again, you can't replicate luck. But the thing that I could say is that I was sort of prepared to pounce when I got a lucky break because so what I would do is I would go to my postdoc lab and I would be there like 10 hours. But the first hour from like 8 to 9 a.m., I would work for myself. So I would prepare myself for like, explore different opportunities, prepare my CV, just like I was working for myself. There was no one there. It was like, and I was getting all my work done. But that hour I was alone, like there was no one in the lab. And I was just working for myself, trying to figure out what to do because I realized like this is not going to work long term. And the not working was not just I don't like what I'm doing now. I didn't even like what success would mean. Success would mean I spent 5 years as a postdoc, and then I got a position where to finance my own salary I had a 30% chance for every grant I wrote in the best case scenario. So I quickly realized like this was not going to work. And so one day I was literally just like looking around and I found the position that I ended up applying for. and you know, I was ready. So like within 24 hours, I had sent an application, and I was very lucky that that all worked out. But at least I was prepared for a lucky break. And again, I don't know that anybody can be as lucky as I've been in my career, but at least, you know, if you're prepared, if something comes your way, you can, you know, exploit the opportunity. Yeah, but that was not an easy decision at all. You know, I thought I was going in one direction, and then I realized, no, I'm super unhappy. I've got to swerve here and go in another.
Yeah, absolutely. Preparations is important. Thanks. Um, Anybody, don't be shy. In the meantime, I have another question, which is, how was your I mean, having left Argentina, you were younger. How was that experience? And of course now establishing your career where you are, how do you look back to that?
Um, yeah, I mean, there's several aspects of it. First, you know, I'm super grateful for the opportunities I've had here in the U.S., which unfortunately in Argentina, it's much harder to do science. So I've been, again, incredibly lucky to have the opportunities I've had. There was definitely a lot of culture shock at the beginning. So one of the things that's very maybe unique to me that's kind of weird is for, you know, personal reasons that don't matter. Like I learned English when I was very young and in a context where I don't have an accent. And I also look a certain way, right? And so like I would have these things where I would ask questions and I would you know, people would look at me like, why are you so dumb? Why are you asking this? Because they thought I was from the US. And you know, I've had questions about like, but is that really your name? It's like, yeah, that's really my name. You know, it's like I've had all these like weird, you know, I'm almost like undercover, right? You know, among the gringos. And so like I had a lot of these experiences at the beginning. But you know, I was also in a city, you know, I spent a lot of time in New York City, which is very diverse. So, you know, I would say it's like overwhelmingly positive. I never felt, like discriminated against or anything like that. I guess I was just super lucky. And it was always, you know, these great experiences of, you know, meeting people from other places and, you know, just learning about them. It's just been overwhelmingly positive here. Yeah.
Can I read one of the, I think it was a bit earlier,
In the chat.
in the
Mhm.
chat,
Sure.
yeah, it says, any AI-driven image analysis platforms that has caught your eye? Also, what do you think of the ethics of using analysis tools whose inner workings are hidden in a black box?
Yeah. So, AI tools that I have used that I found very impactful. I think that the one that I'll give a shout out is CellPose. This is a well-known cell segmentation algorithm. Um, I think it's, it has worked really well in applications that we've done in the lab and in papers that I have collaborations with. So that's one that I would say, you know, worth trying out. And it also has a very low barrier to entry. Um, as far as the ethics of it, I think this black box thing is a real thing. I think it's also a real thing when you have people analyzing things manually, quite frankly. I think that to me, the thing that would be nice to do is I think the aficionados call it containerization, which is, you know, you have the environment in which the analysis was done, along with all the software in which the environment in which the analysis was done, contained in some environment that you can then replicate, so that then people can take that and replicate at least a minimal example of what you did. But yes, I mean, there's this fundamental unknowability about what's going on in those models that, you know, it's yeah, I'm not worried about the ethics of it. I'm worried about the reproducibility of it. And so that's the containerization side, I think, is a good solution, but it's certainly not something that's widespread at all. You know, people are not doing this or even providing minimal examples, but I think that's one way maybe out of this thicket of like, what's going on? How do I trust this, right? You know, we could have another whole talk about, you know, this, but yeah, that would be my sort of 2-minute answer to that question.
Excellent. Yung is saying that your talk was fantastic. It helped me remember how important my role as a core staff member is. Li Yuan as well. I
I
think
think
Elke
someone
had
had
to,
raised their hand, maybe.
yeah, but she just wrote that she had to leave.
Okay, all right.
Oops. Any other questions? We have 3 more minutes. If anybody has a question. If not, well, we want to thank you a lot once more, Pablo. It was wonderful to hear from you. And yeah, for everybody else, please stay tuned to the next talks. We have another 3 guests in the coming weeks. Not next week, because there's the BINA Congress, but after that.
Okay, well, thank you very much, and thank you to the interpreters. Hopefully I spoke at a reasonable pace. But yeah, thank you so much for interpreting all my ramblings. So appreciate it.